have been able to join us. And our lecture today is quite brief, but also a very important lecture. And we're continuing the lecture series on gram-negative bacterium and in essence, gram-negative rods. And today we are going to look at vibrio, we're going to look at Campylobacter, and also we are going to look at H. pylori. And depending on time, we can have a discussion much later. So hopefully you can see the slides and we can move on from there. But as if you've been to any of my lectures previously, I always like to show this slide, just drumming on the issue of what you're facing right now, that we are all saying this is actually the real pandemic, where we are seeing fast antibiotic resistance or antimicrobial resistance dominating most aspects of medicine. And this is a major concern, as I shared with you earlier, from WHO and from other organizations. They're saying, again, by 2050, we'll have virtually no antibiotic or antimicrobial working. So you can imagine at that point, having even a simple surgery, a simple toothache or cough that is bacterial strep throat will be quite difficult to treat. And the infections we're going to look at again today, we should bear in mind what may be fueling them again. It could be antibiotic resistance. And in this article, as I mentioned last time from the New York Times, it was depicting in Kenya where we have a chemist, maybe unlicensed, at every corner of the streets in the slums. They did this study in Kibera, where someone would turn up at the chemist and ask for a simple medication. And they had some infection, and the first thing the chemist would give them is an antibiotic. So really, we just want to make sure that as you're going through microbiology and your fourth, fifth, and sixth year, we are raising this desire need to be antimicrobial stewards or antimicrobial stewardship, where you're making sure that we're only treating using antimicrobials where we have done correct diagnosis and identified the causative agent. So this is something I always like starting my lecture series in. So today we're going to look at vibrio, and the pathogen of interest here causes cholera, and it's a major infection around the world. And here's just to show that they've even made movies and documentaries on cholera. I found this movie quite interesting, Love in the Time of Cholera, looking at some interesting couple before the world wars, and how cholera disrupted their lives in London, which again had major epidemics at some point in the early 1900s. And up to today, we still see some reports that in certain areas, there's been a cholera outbreak, and this has been quite a major concern. And it's something for us to think about, even in Kenya, where we saw a few years ago in Kibra, not just in Kibra, in high-ended, high-upmarket resorts or restaurants such as Weston, where some of my colleagues even had some lunch there, and by the evening they all had cholera-like symptoms. So this is not just a disease of poverty, it's a disease that we see where food or you consume items that may be contaminated with the vibrio pathogen, which is our major focus today. So our learning objectives by the end of this lecture, we're looking at these gram-negative rods, which is the basis of my slides or my background today. And we're going to look at vibrio cholerae, and we're also going to look at other microaerophilic members such as Campylobacter and Helicobacter and Aeronomus, which all fall under this category of gram-negative rods. So our pathogen of interest falls under this family, the first one, that is vibrios, which belongs to a bigger family known as Vibrionesae. And the major theme of the last few lectures, we've been looking at gram-negative rods, and they're quite characteristic because they have like a comma shape, as you can see in the background of my slides, where it looks like it has a hook, but this is very important for this pathogen because it's able to penetrate and dig through or burrow through the intestines, the mucosal layer that lines our intestinal, our gastrointestinal tract, to be able to leach on and obtain their nutrients. And it's through that we see the different pathogenesis or different symptoms a patient may have. Again, as you can imagine, they need to dig through or burrow through this major barrier, this is the mucosal layer. They need to be highly motile, so they have a single quite powerful polar flagellum, which is a virulent factor which assists us to move and be able to be able to attach itself to different intestinal cells to obtain their nutrients. And again, it has different characteristics of spore forming. It's not capsulated, so it relies on other mechanisms to be able to resist and counteract the host immune system, which the capsule, as you've studied before, protects the pathogen from phagocytosis and other immunological responses. So it's not capsulated, but relies on other factors such as the antigens and toxins to be able to burrow through, attach and resist the host mechanism that is mounted by the immune system. The anaerobic, facultative anaerobes, and they'll thrive in regions that have less sufficient oxygen, and something again, they possess both H and O antigens, which you see the importance of these factors in the pathogenesis of this pathogen. Sorry? Okay, I thought there's a question being asked that assists in its virulent factors to help be able to settle in the host gastrointestinal tract and retrieve nutrients, reproduce, and resist the immune system. So here, for us to be able to identify it properly in the lab, which is key in diagnosis, we know that what we've seen in the previous slide, it's highly motile. That's gram negative. So if you're doing identification, that's an important factor. It does not take up the gram dye, and we will not be able to visualize it. So it's gram negative, and the patterns, again, when you do your practicals, will be quite obvious to you. Again, you've seen that it has a characteristic rod that it's curved or comma shaped with a single powerful polar flagella, which assists us to burrow through in a circular motion so that it can overcome the mucosal layer that lines the intestines and the gastrointestinal tract to be able to attach and retrieve nutrients as it needs. Other important factors in this early parts of the lecture is, again, really key for us to help us identify is how do we visualize it. So we've looked at factors and how we see it under the microscope. But when we culture it, once we get your stool sample, we'd want to also culture and use different culturing techniques. We can use the McConkey agar. We can use more specific culturing techniques such as the TCBS, which again, you'll be able to visualize and work with it in the lab when you come for your practicals, where this is the TCBS. And when we culture specimen that we suspect is Vibrio cholerae, we should be able to see this mucoid yellowish colonies growing. So another factor that may assist us in identification, which is key in diagnosis, is that it grows in the presence of no salt. So when we are preparing the agar in the lab, we make sure that particularly Vibrio cholerae will be able to thrive in salt lacking agars. A good something for you to have in the back of your mind, particularly if you want to differentiate with other vibrios. Remember, it's a large family, so other vibrios that are not cholerae are halophilic, so they'll be able to thrive in salt-like conditions, and this is what we call halophilic salt-loving vibrios, and you'll be able to find them also in seawater and or in regions where are salt-rich. So again, an important factor to have in the back of your mind when we come to do diagnosis. So moving on, as I mentioned, there's Vibrio cholerae, which causes serious pathogenic consequences and the symptoms you're going to see as we continue the lecture. And the other vibrios that are halophilic, and you find them thriving in salt-like water. So if you like your seafood, like, things such as shellfish or prawns or things that you find in the ocean, and they're not well prepared, they may predispose you to the other Vibrio family that also cause some gastrointestinal tract disturbance, such as Vibrio parahemolytics or Vibrio vulnificus, which again are halophilic and are salt-loving. So that's important for us to know if you're not certain of the Vibrio that you've identified in the lab. You've done your gram stain, your patient has those gastrointestinal tract symptoms of that characteristic diarrhea, and you've done your gram stain, you've done your TCBS culture, a different way to be able to differentiate the different subspecies. If you don't have molecular techniques, is to expose them to salt-like conditions and do another culture, subculture, which will then help you differentiate between Vibrio cholerae and the other Vibrios within the subspecies. So where do you find out this Vibrio? And again, the way I've introduced you, I've introduced the Vibrio family, I've introduced you in simple words. Again, you're going to look at that, the diagnosis, how do we identify them? Because that again is key in our patient management. We've looked at, in just a summary form, the microscopy and also culturing conditions that will come back to the lab diagnosis again at a later time point in this lecture. So let us look at where we find the Vibrio species. As I mentioned, you have to have consumed something that is contaminated, such as food that's not well prepared or contaminated water. And of course, if there's increased salinity and optimum conditions, you may predispose to other Vibrio species, such as the hemolytic Vibrio. And as I mentioned, if you consume seafood such as shellfish, you'll see that if it's not well prepared, you may predispose yourself to some of the Vibrio species. And there's some literature, some work that are saying this could be because there's a symbiotic relationship where they get some nutrients from these sea creatures, and the shellfish also get some benefits from having that symbiotic relationship, and they're a major reservoir for the other Vibrios. And again, another reservoir where you may get Vibrio or get contaminated or get the infection, if you had someone preparing food and they may be infected with Vibrio. They could be asymptomatic, but they may be able to pass that infection from one person to the other. So this has also been a route of transmission that has been shown in literature. And it's important to note here that asymptomatically infected humans may also serve as an important reservoir and may pass on the infection from one person to the other, despite showing no symptoms at all. So here we see that Vibrios from contaminated products, foods, or water, again, and also people who act as reservoirs have no symptoms whatsoever, but they can pass this infection to the next person. And again, so it's very important, if you don't know your food handler, make sure that the public health acts, make sure that people are working in hotels and other food preparation areas have the correct certificates, correct vaccinations, so that they don't pass this infection from one person to another. So I've mentioned cholera, which is a major disease or infection caused by Vibrio, particularly Vibrio cholerae. And as you know, it's a gastrointestinal tract infection and causes quite severe diarrhea. And one thing about the diarrhea caused by Vibrios, it looks like rice water. So for you who prepared mchele for lunch or whatever, and want to wash it, and you are washing your rice, you see the water becomes whitish creamish in nature. And the cholera diarrhea has similar characteristic that looks whitish creamish in nature. Very important to have that at the back of your note, because also that helps in patient management in terms of diagnosis. When you do your macroscopic investigation, which means what do you see with your naked eye, which is the beginning of your patient management. Once you've taken the patient's symptoms, you look at the samples, you look at the diarrhea, what color is it, and if you spot some whitish creamy, if it's whitish creamish in nature, again, that would help you suspect that your patient may be actually infected Vibrio cholerae. I know we're talking about diarrhea and all, and it's just a few minutes to lunch. So again, forgive me for that, but that's the nature of the lecture today. And what causes this characteristic color? It's because of the toxins that it produces, and in particular the cholerae toxin, which again burrows through or digs through the gastrointestinal tract, the mucosal layer of the gastrointestinal tract, which again erodes it, and some of that erosion gets into the fecal matter or through the intestines, which then is eliminated with the watery diarrhea that you will see. So the toxin plays a big role in the pathogenesis of Vibrio cholerae. So again, we are still looking at Vibrio cholerae, and I need to mention that apart from its characteristic diarrhea, it's categorized as a category B bioagent. So if it falls in the wrong hands, like terrorists, or right now where you've heard of course, you've heard of the Russia-Ukraine war, and the Ukrainians are quite afraid that the Russians may use bioweapons, and one way they can bionize cholera toxin is to pour it or introduce it to a major water supply, and that can really weaken your enemy or the regions you're fighting because of the severity of the diarrhea. So it's categorized by the CDC as a bioterrorist weapon. So again, for those who are researching or working with cholera, they need to be in a level 3 lab where they're wearing enough protection, and in a level 3 lab or level 4, again, the biosecurity is key on who is handling this pathogen in terms of their research and how is it being stored. And again, I've mentioned how is it introduced. It's through ingestion of contaminated products, and poor sanitation is a key source of outbreaks, not just in our region, but other areas where we've been seeing outbreaks of cholera caused by Vibrio cholerae, which is a major pathogen in this to cause this infection. And where have we seen this infection? We've seen it in Kenya, I think in the last 2 years or so. Again, COVID made us not forget, but we were not able to highlight other infections that may be circulating. I saw another research article that was showing, because we were very careful with hygiene, washing of our hands, and also careful in what we are consuming at the early part of the pandemic, we were able to see a drastic reduction of gastrointestinal tract infections. And again, really showing how hygiene and proper sanitation is key in prevention of some of the severity of the diarrheas and other gastrointestinal tract that we're discussing today. But where have we reported cholera outbreaks? It's in all parts of the world. Early on, before the World War I and IIs, even in the more industrialized areas, they were able to see cholera, but at the moment, because they're able to take care of their sewage system and all, and their water treatment plants, it's rare to hear of cholera outbreaks in the Western world, but we still see it in India and Sub-Saharan Africa. So again, it's still a concern in our region, and that's why we are gathered here today, so that we can see what causes cholera, how do we diagnose it, and also how do we treat. And again, that's just a continuation of the slide, where in the last 5 years or so, cholera outbreaks have been reported. Of course, Kenya is in red, so we've had severe cases of cholera, and not just in the city areas or in the slums, but also in the refugee camps, in Dadaab and other areas where we have a high concentration of people living in one place, and there's poor sanitation. And this is where we see these outbreaks, and this you'll see the patients or the samples coming in from, and it's proper for us to be able to understand how to diagnose and treat such outbreaks. And again, as I mentioned, Southeast Asia, where we've seen reports of this infection. But in the last 5 years, we've not seen major reports of cholera in the Western world. But now with the situation as it is in Ukraine and in Eastern Europe, we may start seeing a high rise of refugees or people living in high concentrated areas. I saw such a report coming from France, where because of the war in the Middle East, in Yemen, most of the refugees kind of move over to Europe. And there's that constant fear of their disease experts that they'll start seeing diseases such as cholera in the refugee camps, and they have a heightened level of concern and investigation. So what has caused the epidemics that we see in Kenya and also in Southeast Asia and in Sub-Saharan Africa? There are two major serogroups, and I know one of you may ask what are serogroups. This is the vibrio cholera itself, but then it has different antigenic structures at its outer cell level or outer cell wall level. And this can be detected when you do a serologic examination, or you introduce antibodies and it agglutinates in different patterns. And this is how we see the different serogroups. And in vibrio case, we see that it has two main serogroups, which are O1 and 0139 serogroups that form the vibrio cholerae family, or some of the serotypes that have been implicated to cause major pandemics or epidemics in India and also in Sub-Saharan Africa. And it's important to note, because I see this question coming over and over again in your MCQs and in examination, name different serogroups of cholerae. So under the O1 serogroups, we have sub-serogroups such as the classical one and the L-TOR group as well, that have been implicated to be quite infectious in causing or fueling the epidemic of cholera outbreaks. So I don't have to drum on how it's introduced the routes of transmission, particularly of these serogroups that I've mentioned, the O1 and 0139. These are the main routes of transmission. So we need to be able to make sure that you've taken control of our sanitation. I know in Kenya we have a huge problem, particularly get really concerned when it rains and you see your drainage system and they get clogged up. And once it clogs up, there's a lot of flooding in residential areas, and this flooding may affect the drainage systems, the sewerage systems. Again, that's why you see that now fecal matter flowing in that flooded area, and this can seep through and in our food system or food chain, and that's how contamination may occur. So that's something for us to watch out for. Okay. And sorry, it's just a small image. I was trying to move to there. As I was mentioning of the sewerage system, flooding system, we like consuming food that may be prepared on the streets. I've seen also an image that goes around in social media where we buy our vegetables. And when you look at where these vegetables are growing, they're growing in Ruai, where we have a huge sewage system, and there's always that fear that is introduced back into our food chain, and that can, if the sewerage system is contaminated with vibrios, may introduce the vibrio cholerae to us in our population. And also I've mentioned high-end restaurants that have been implicated. I remember also, apart from Weston, there was a Grand Regency when it used to operate. A conference happened there, and by the evening, majority of those people who attended that conference had vibrio cholerae or had cholera diarrhea-like symptoms. And again, I remember, as I mentioned, my colleague in the evening had already been admitted because of the severity of the infection, and he must have received a huge bacterial load, so it was highly transmissible for him to have consumed something at lunch hour, and by evening he was already admitted in hospital, and the treatment had already been implemented. We also have this issue of hanging latrines. I've not seen it as a major issue here in Kenya, but in India, this is something of a major concern, where people relieve themselves in flowing water or in lake regions. And this again, there's a contamination, introduces it back to the populations. Again, we're interested in public health and public sanitation. These are areas of major concern that we need clear policies or guidelines, and to be able to take control of those routes of transmission. And unfortunately, we get to consume a product that is contaminated. We look at now the pathogenesis of that infection. I've already mentioned some of the virulent factors, and one of them, the bacterial, has fimbriae that helps it attach to that mucosal slimy area of the gastrointestinal tract. So it needs to be able to adhere to the epithelial cells through the pili, the fimbriae, and of course, with its powerful single polar flagellum, it's able to burrow through, so it can attach itself once you introduce it or you consume a product that is contaminated. Another thing about Vibrio, it's able to survive the very harsh conditions of the gastrointestinal tract. And what do I mean? It has high levels of acidic conditions because of the hydrochloric acid that the stomach itself produces, that can be able to assist in the digestion. So it's able to have factors that neutralize that acid, which is very important for it to be able to survive and cause a serious pathogenesis that we see. Other factors that the bacteria needs to have, virulent factors, it has the enzymes and the toxins, enzymes such as proteases, or which in textbooks it interjects that with mucinase, and that helps it digest the mucosal layer. Very important for it to have these mucins so that it can penetrate and be able to attach to those epithelial cells. Important for it so that it can retrieve nutrients and other factors to help it thrive. And it's with that we see this characteristic symptoms. So I've mentioned the enzymes, the structural virulent factors, and also the toxins that it produces, and of course the flagella. And the major toxin that we need to focus or know about is the enterotoxin, cholerae toxin, or in simple words, CT, which again is important for it to be able to digest through the mucosal layer together with the proteins such as mucins or mucinase, so that it can be able to burrow through the different epithelial cells. And I've mentioned the cholera toxin not only helps it in the attachment and burrowing through, but also it changes how the absorption of nutrients that occur within the gastrointestinal tract, particularly the intestines, that it's able to inactivate very important mechanisms such as the G protein. And once it inactivates the G protein, because the G protein helps in the closing and opening of different channels so that the body can absorb nutrients, the body can absorb also water from the food that is consumed, but the toxin interferes with this mechanism in that the body is unable to absorb these nutrients and also the water that the person has consumed. And once these G proteins and other mechanisms of switching on and off are interfered with, and other factors such as CAMP, which then because of the different interference, we see some proteins and factors upregulated, will mean that water has not been absorbed properly by the body, and it's passed on again to in the stool or in the fecal matter, leading to the characteristic diarrhea. And it's because also of the burrowing or digging through of the gastrointestinal tract, not just by the toxin, which it interferes with the mechanisms of nutrients absorption, but also the proteinase or mucinase, which helps in the digestion or breaking down of the mucosal layer, which again is released in the fecal matter. So we see the characteristic whitish diarrhea that again is important for us to help us identify cholera diarrhea. So again, I've mentioned the toxins, which interferes with how the intestines, how water is being absorbed, and also not just water, but other micronutrients, salts, and items such as sodium. And if this mechanism is interfered with, the body is unable to absorb these nutrients and also get hydrated. So your patient will be severely dehydrated and will have this characteristic sunken lips and sunken cheeks that I'm going to show you in the later slides. And it's because of these mechanisms that the cholerae toxins interferes with, and so loss of cell nutrients, which again leads to the characteristic diarrhea, together with the interference of the integrity of the epithelial layer and the mucosal layer, which then again feeds into the fecal matter or diarrhea. And that's why we see a characteristic, a very unique type of diarrhea. So some of the symptoms, it's quite highly infectious, a simple small dose, high infectious dose, such as the bacterial load that we see here, can lead to quite severe symptoms. So again, it's quite infectious. A small dose can cause severe symptoms that a patient may have. And depending on the serogroup, you'll see that different serogroups needs just maybe 1 or 2 or 3 bacterial load to be able to see the symptoms. And we saw that in the classical and the LTOR subgroups or sub-serogroups of cholerae. So we see an abrupt onset of symptoms. We see the vomiting and quite a watery diarrhea. And if you're measuring, your patient may meet even up to 15 to 20 liters a day. And this really drastic fluid loss that I've explained that is fueled by the toxins and other factors may be quite fatal to your patient if you don't initiate dehydration mechanism or hydrating your patient at the onset of symptoms. Another very important characteristic, as I mentioned, is this rice water like diarrhea or stool that is usually colorless or odorless. When you look under the microscope or you do your biochemistry, you not see any of the proteins, and it's normally speckled with mucus, again, because of the burrowing or the digging through of the mucosal layer. So people most at risk, of course, anyone who's consumed anything that is contaminated is at risk, but also from research, you see children who have a less developed immune system, and also because of their vulnerability, children and the elderly are quite vulnerable. And again, it's because of either low gastric acid levels, so the bacteria is able to thrive and reproduce even faster. And also, I've seen research that is showing, though implicating some certain blood groups may be prone to this person being more susceptible to severe cholera symptoms, severe diarrhea. And the hypothesis here was the bacteria, because of the, particularly people who have O blood group, the bacteria is able to adhere better to the gut lining compared to the others. That is still something under investigation. So diagnosis, I've mentioned there are already sunken eyes and cheeks because of the severity of the dehydration that may occur. The skin also loses the, that glowy like characteristic. So the skin turgor is quite decreased. It'd be less urine production, again, because of the drastic loss of fluids through the diarrhea. The watery diarrhea, as I mentioned, they'll be able to see fluid without any red blood cells or proteins, and have a high level of electrolytes because they've been lost, and also an enormous dose of the vibrio cholera itself, which again, if the sanitation is not quite good, this can be passed on to the sewerage system or contaminate other products that someone may consume, and hence fueling this epidemic. And as I mentioned, the diarrhea has quite a white water, rice water-rich appearance, which is key for us who are trying to diagnose and treat this diarrhea and use the appropriate measures to prevent cholera. So these are things that we need to be aware of, visualize as we are managing our patient. So How do you make the diagnosis, which is key for anyone doing microbiology? Make sure you're collecting the correct specimen. For us here is a watery diarrhea. And if you're in an area that's not near the lab, how are you transporting it? These are key points for us in lab diagnosis. We normally test in this as well in exams, in your MCQs. What are the factors that we need when we're doing laboratory identification? So a specimen, I want to see you identify the specimens correctly. How are we transporting it to the lab? We normally use the Carrier transport medium. Where is it important for us to be concerned about how we are transporting it? Because we don't want the bacterium to die off in the middle of transportation, because we want to be able to diagnose and identify that bacterium. So we need medium, transport medium that has optimum conditions, has enough nutrients that we can maintain the bacterial load in that specimen. And once it's in the lab, before we do microscopy, we have to do the macroscopy. I mean, what do we see in our with our naked eye? And then we may want to use enrichment medium. We cannot make the diagnosis fast enough. After the microscopy, we can use alkaline peptide in water to be able to enrich the growth of the bacterium to help us identify. And once you do the microscopy, you may want to do your subculture, you do your culturing, and you can use the TBS culturing conditions and or the agar, and where we be able to see a mucoid yellowish colonies. And you may want to repeat the microscopy and do other biochemical tests such as the oxidase test. And again, all this will be able to demonstrate to you much better in the laboratory. So note this, the TCBS in your spots of practical exam. I've seen questions where they have this displayed, and of course, without identification, we ask you, what are we trying to identify here? And if you get that right, the next question would be what infection it causes, or what tests would you do to confirm that it's cholera or the pathogen that you identified, or treatment options. So when you're approaching your spot exam, make sure that you are not only able to identify the pathogen or different testing techniques, but you know, once you've identified the pathogen, how do you treat it, or what is its pathogenesis? So again, I'm sure in year 2, we introduced to spot exams in microbiology, so this should not be something very new to you. So again, I've mentioned the TCBS, that stands for this, and we're using, if you're trying to identify it, other vibrios such as parahemolyticus, you'll see different type of colonies. And again, that is influenced by the uptake of the salt that may be found. Other tests, and for me, I always prefer the molecular test because it's pretty fast, very direct. Within an hour or 2, you have your test because you're detecting the DNA or the molecular sequence of that pathogen. So you have that capability, that's fantastic. But there are also rapid tests such as this, which again, you just dip this onto the specimen or the stool, and if it turns out positive, or the 2 lines, again, you can be able to quickly identify the source of the epidemic and start off treatment of your patients. Okay, so treatment here, it has to be quite instantaneous or immediately, because again, because of the severity of the dehydration, the patient may succumb. So make sure your patient is well rehydrated. You can do even intravenous rehydration and then initiate antimicrobial therapy, bearing in mind the different antimicrobial resistant patterns or the antibiogram that your hospital, your clinical setting has prescribed. And how do we prevent this? Of course, consume your food well. If you're traveling, particularly in areas that you're not very sure of the food, make sure you try and consume food that is well prepared. And I saw this as in some of our public health website. If you boil it, cook it. If you can't boil it, cook it, or peel it, then forget what you're consuming, which is key. Of course, sanitation is key, and of course, the vaccines that are now available and that should be given to anyone who is handling food. And there's also usually food handlers inspection that is important. So they have these vaccinations and have them given correctly. At the moment, I heard of cases where there's some corruption in some restaurants or some food handlers who come into corruption ways with either city council officials or now NMS, and they get issued this food handler certificate without proper vaccination. So these are things us on the ground and you who will be our champions in public health, make sure that people are knowing why they need these vaccines. And I think knowledge is power. They won't have to have those shortcuts and would have the vaccines given to prevent the breakouts that we see in even high-end restaurants or facilities. So make sure there's good food handler certification even in these places. So for the class, I'd want you to read more on the cholera vaccines that are available. There are now newer vaccines that are coming up using the mRNA technology that we saw in COVID. And we want to make sure that once you give this cholera vaccine, it elicits lifelong protection. And that will be key to help us eliminate and eventually eradicate cholera from the world. And of course, I want you to also read more on antibiotic resistance patterns in Vibrio, and not just Vibrio cholerae, but the other Vibrios of interest, which again should form part of the notes that you read through at your quiet time. So let us move on to Campylobacter, which is also a gastrointestinal tract. And the theme here is this gastrointestinal disturbance, as you can see from these drawings here. And our pathogen of interest has two single polar flagellum. And now you appreciate why it needs this flagellum. It needs to be able to penetrate through the mucosal layer and attach itself to epithelial cells to obtain its nutrients. And for it to be able to surpass even the harsh conditions of the intestines and also the peristaltic movements of muscles of the gastrointestinal tract as it's digesting food, it needs quite strong or powerful flagellal movements so that it can resist the host mechanisms and be able to retrieve its nutrients. So Campylobacter, it's another gram-negative rod, and it looks like seagulls. So the textbooks will always say it has a seagull appearance, and motility it needs to be able to have this polar flagella that is on two polar ends, and it moves in a darting-like movement. So like, if you can see my finger movements, so it needs to be able to move in that manner, so that it's not just darting through, but burrowing through or digging through the gastrointestinal tract, that can penetrate and colonize in such mucosal environment. And Campylobacter are the major species here, particularly for those who like poultry products such as chicken. They're quite prone or have been known to be reservoirs of Campylobacter jejuni. And if you don't prepare your poultry products in particular well, you may be predisposed. I remember I took a trip to China a few years ago, and Chinese, it's quite interesting how they consume their food, in that their starters was chicken, but cold chicken as a starter. And in my mind, all I was thinking about, wow, Campylobacter. And I think being a microbiologist or studying these pathogens for quite a while, you get quite over concerned about certain aspects, and I did not touch the cold, nicely arranged breasts, chicken breast and chicken thighs, because I was quite afraid of them being contaminated, them not being well prepared, and predisposing myself to Campylobacter. So if you're looking at it under the microscope, again, you'll see that curved nature that it moves with a darting corkscrew-like fashion. So source of Campylobacter, and the major one being Campylobacter jejuni, is commonly associated with poultry. That explains why I could not see myself consuming cold cuts from chicken. And it's known to be a reservoir because it's a part of the natural flora or microbiome in poultry. So if you consume not very well prepared food products, you may predispose to Campylobacter digestive issues such as diarrhea and gastrointestinal pain. So once you consume something like that and it colonizes you, you have such characteristic diarrhea, etc. And from your patient, you need to collect stool. again from the diarrhea. You can have that also brought in the lab. You can also have a blood specimen, which helps us look at the antibodies, or if your white blood cells have been elevated, show that you've been infected. And what we do a wet mount, where we make sure that you're able to, first after doing macroscopy, you're able to visualize it well, and under the microscope, you'll be able to see the darting mortality and the curved like rods that you've seen. And we normally use Quirose medium, a specific commercially available medium, which again is quite important for Campylobacter and nutritious enough. And we normally culture it for around 2 days or 48 hours and at quite optimum temperature. I think you remember from your biology, high school biology TC, where we saw that the body temperature of poultry is around 42 degrees. And this is the optimum condition you need also for Campylobacter. And that's what the conditions we use when you're culturing it. And it's microaerophilic, so it needs a copious oxygen amount to be able to. Seem to have lost my connection, but I'm back now. So I was explaining about the Quirose media and also microaerophilic conditions, which are conditions that are, and I remember from your introduction to bacteriology, are microorganisms that require oxygen to survive or to thrive, but they can also survive in lower amounts of oxygen, and that's from what we find in the atmosphere. So they require just a little bit of oxygen. They can thrive in areas with less oxygen than what we find in normal temperatures or normal atmospheres. So that's what microaerophilic conditions are, and hopefully that was covered in your second year or when bacteriology was introduced. Remember, there are gram-negative rods that look like S-shaped or that have a curved shape, and most of these gastrointestinal pathogens are normally oxidase positive. So how do we treat Campylobacter diarrhea? After taking a patient history of the diarrhea, this time is normal diarrhea, watery diarrhea. You take the stool sample, do your investigation. You can, as I mentioned, under the microscope, or you can use molecular technology. And once that is diagnosed, make sure that your patient is well rehydrated to avoid severe loss of nutrients and fluids. And you can initiate antimicrobial therapy, such as erythromycin, but make sure that, again, you're watching out for antimicrobial resistance. And as always, for any diarrheal symptoms, we discourage use of antimotility agents. So let the diarrhea run its course. Most of the time it's self-limiting, because that helps also eliminate the pathogen from the patient. So that's important. Just make sure you're watching out that your patient is well hydrated and they see with severe symptoms, or you're dealing with a very young person or old who is more susceptible. Make sure that we are properly administering antimicrobial therapies. So moving on to one of the last aspects of this lecture, that is Helicobacter. And these Helicobacters have gained quite a bad press in Kenya the last few years or so, because it seemed that everyone who had a gastrointestinal issue that was long-lasting, they would get treated for Helicobacter. And we had an issue with that because were they doing the proper testing? Were they doing proper diagnosis? Probably not. I even remember a child who was around 6 months who had some gastrointestinal issue, and the doctor quickly gave the diagnosis to the mom that the child has H. pylori and they needed to go into treatment. And that, again, is something we strongly refuted, because at that age, again, there could be other factors that may be at play and not H. pylori at that point. So let us look at H. pylori, and we cannot look at H. pylori without mentioning some of the Nobel laureates, such as Barry Marshall. I don't know whether you know his history. This was just a few years ago now, where this guy was a doctor who kept seeing his patients who would complain of gastrointestinal disturbance, and most of them had stomach ulcers. And at that point, we're treating ulcers because I would say the ulcers are probably associated with anxiety. Are you anxious? Are you depressed? But he realized there was a pattern with these ulcers, with gastrointestinal disturbances, and he had a hypothesis that most likely is being caused by H. pylori. And you'll see that H. pylori thrives inside the stomach. Very few pathogens are able to do that because of the harsh conditions, but H. pylori can, because it produces factors that neutralize the acid. So what he did, he actually consumed or drank solutions that had a high concentration of H. pylori. And of course, he got the stomach ulcers, and he had those gastrointestinal conditions or disturbances. And if he wants to develop those conditions, he prescribed himself or treated himself with antibiotics, and quickly the ulcers resolved, the gastrointestinal issues resolved. And with that, which is quite unconventional, he was awarded the Nobel Prize in medicine because of his discovery of linking stomach ulcers to H. pylori and not to depression or other mental disorders that previously the scientific community was linking that to. So yeah, well, kudos to him, but quite unconventional way of proving something. So of course I had to mention him. So what is H. pylori? The normal flora of the stomach as well. And once if you have someone diagnosing you with H. pylori, we have to also consider what's the bacterial load. Is it high because it's one of the normal floras that colonizes the stomach, some part of the microbiome, quite contagious. And up to today, we are still trying to investigate what is the route of transmission. Is it eating contaminated products, or someone who has a natural high bacterial load of H. pylori prepares your food even without the symptoms? Can they pass this infection from one person to another? We don't know. It's something under investigation. But main risk factors, once you have a high bacterial load of H. pylori, apart from the stomach ulcers that you see, and this is because of the acid nature of the gastrointestinal tract, and we're going to see how that comes about and how it's able to cause those ulcers. So main risk factors of having high levels of H. pylori, apart from the ulceration, it can also lead to stomach cancers and other complications in the gastrointestinal tract. So it's important not just to treat, but be able to make a diagnosis. Again, there are, as you mentioned, the characteristics, they're gram-negative, microaerophilic, so again requires oxygen to survive, but not at high levels. That's what it classifies microaerophilic. They're quite motile, and they need to produce all these enzymes so that they can first of all burrow through or dig through the gastrointestinal tract. They need urease to be able to hydrolyze or neutralize the acid that is found in the tummy or in the stomach, and also they produce oxidase. And you can see here from this diagram on top of my head, you can see it has quite a high level of flagella, which assists in its movements. So virulent factors, as depicted here, and I've already maintained, has a high motility rate or movement because of the copious amount of flagella that it has, which needs to have that so that it can burrow through the stomach lining, which again is important for it to be able to penetrate, has different proteins such as adhesions, which helps it adhere to the epithelial layer of the cells. And of course, you've seen the importance of mucinase, which again degrades the stomach mucus, the mucus lining. And so you can imagine if it's degrading the stomach mucus, which protects the stomach lining from the hydrochloric acid. Again, the hydrochloric acid gets in touch with the layer of the upper parts of the stomach or the inner parts of the stomach, which was protected by the mucosal layer, leading to tissue damage. And that's how we see ulcerations forming. I've already mentioned urease, which again helps neutralize the local acid environment for it to be able to thrive. But of course, that also leads to the tissue damage that we see that leads to ulcers. And also, of course, it produces other factors and other toxins, which helps penetrate into the epithelial layer. And this again, I'm sure I'll be able to share these slides with you for you to be able to see other important virulent factors that H. pylori needs to be able to thrive inside the stomach lining, which again, I've mentioned, it's quite harsh because of the acid layer that, or the acidity that is found in that region. So I've already mentioned to you the pathogenesis. It needs to be able to attach itself to the mucosal layer. It needs to produce urease, and from urease, it's able to hydrolyze the hydrochloric acid where ammonia is produced. So again, this is detrimental to the tissues around there and leads to the damage of the tissue lining, leading to gastritis, peptic ulcers, and if not treated correctly, can predispose your patient to stomach cancers and other complications. So again, clinical manifestations here, I've listed them from that acidity, the pain, loss of appetite, sometimes bloating because of the copious amount of ammonia that is produced, gassing and bloating-like nature. Again, these are all characteristic symptoms that you may be infected with H. pylori. And of course, I've mentioned you may be also predisposed to gastric cancers. How do we make a proper diagnosis? This is always the bone of contention. There are several lab testing, such as rapid test, dipsticks. You can test for ammonia or the gas that's produced because of the hydrolyzation, and you can do some carbonurea breath tests, which are also important in lab diagnosis. And you can also do cultures in the lab and other testings, such as gastric biopsies, which again helps us identify H. pylori and initiate treatment immediately. So treatment, there's quite a hybrid therapy that combines triple therapy to be able to reduce the levels of H. pylori in the gastrointestinal tract so that the patient may not progress further with those ulcerations and be predisposed to stomach cancer. So we have an example here. This is given within our country, meclanaxin. There's also levofloxacin, that's triple therapy that can be initiated once you make a proper diagnosis, but also watching out for the symptoms of your patient. So a summary of H. pylori is a common precursor of gastritis and peptic ulcers and a risk factor for gastric carcinomas. And I thought this is really interesting in that H. pylori produces these factors. I've mentioned the mucins, the mucinases, the ureases, which again neutralizes the acidic nature of the stomach, allowing this pathogen to thrive in the host. But again, these factors degrade the mucosal lining and the tissue, leads to tissue damage, leading to more peptic ulcers and other complications. And you need triple therapy, as shown here, which again is able to counteract and effectively assist your patient from recovering with H. pylori. So a nice cartoon which I really like that summarizes H. pylori and how to treat and also diagnose it. So last part of the lecture, when I mentioned that we're going to look at Aromonas. These are just 2 slides. That is a newly emerging pathogen. Again, we need to be watching out for anything that's newly emerging or reimagined, because we've seen with COVID and the coronaviruses, the level of pandemics they can cause, because again, our system, our bodies have not seen these infections before, so we don't have a naturally built immunity, which again can cause the pandemic nature. And for aeronomas, has been shown to have an zoonotic approach, in that it can be spread to humans through the pets and wild animals, and again, eating foods that are contaminated and can lead to all these complications. And again, with aeronomas, I would like you to read more on these newly emerging pathogens and the different characteristics. Again, they're dealing with gram-negative rods, which are quite highly motile, the different significant human subspecies that have been identified so far in the Aeromonidaceae family, and where we are more predisposed to. So that's something I'd like you to read at this lunch hour, or when you're reading through, and other important clinical syndromes that are important for us to be able to make a proper diagnosis. So in conclusion to this lecture, I know it's lunchtime now, we've looked at H. pylori, Campylobacter jejuni, and of course, we've spent time looking at vibrio. They're all gastrointestinal tract infections. These are gram-negative rods, which have quite unique characteristics. Either they're S-shaped, they have very powerful flagellum, and this flagella is important. They produce toxins and enzymes which degrade the mucosal lining, and which may lead now to those gastrointestinal disturbances that we've seen, and other important symptoms that we need to take key note of. And of course, they're highly contagious, and we have levels of causing epidemics such as vibrio, and that's why we need more studies. I've seen organizations such as the Wellcome Trust and CDC giving money specifically for us to be able to diagnose and treat and prevent Campylobacter jejuni, and on which areas are more prone to within our country, Kenya. And these are, again, these are infections that you need to be able to understand so that you can prevent them. So allow me to stop there. I welcome any questions. I've seen many of you have exited. Been lunch hour, so you can type your questions, and allow me to stop there. Thank you. Okay, so I see questions on the Zoom chat box. Should asymptomatic H. pylori be screened and treated? The problem is, if we screen everyone for H. pylori, and we've seen that H. pylori is one of the microbiome or normal flora of the gastrointestinal tract, we may then put everyone into treatment. So what we would agree on first, let's look at the symptoms. Are they asymptomatic? If they're symptomatic, maybe it's something of watching and waiting. But if we see characteristic symptoms, gastrointestinal pains and disturbances, then we may be forced to treat so that we can prevent the pain, the gastric ulcers, and also consequently, we don't treat that, it predisposes patient to gastrointestinal cancers. So that's something we need to watch out for. What does CFU mean? CFU is a when we're counting bacteria in culture, or we are trying to identify the concentration of the microorganisms, we normally count and call that colony forming unit, CFU. And again, I believe this is something you may have done in the introduction to bacteriology. And with the CFU is very important for us, not just for bacteriology, but also fungi identification, because it's a CFU that we need to be able to measure and estimate the number of bacteria or fungal cells in a sample and determine how severe the patient may have his symptoms or his symptoms. So with that, those are the questions I'm seeing. I thank you. Thank the class, even the ones who remained past 10 minutes past lunch hour. Enjoy your lunch, and also want to take this opportunity to wish you a blessed Easter. Thank you very much, class, and see you soon.
Thank you very much, and have happy Easter.